Hi, welcome to tonight's lecture on Chapter 7. We're going to do aerobic respiration and fermentation this evening. I will tell you that Chapter 7 in your textbook has tremendous detail, way more than I would expect you to know on this topic. So I would encourage you to watch this video once, maybe twice, maybe 3 times. Print out the notes that I'm writing and see if you can follow it along when you re-watch the video. There are some diagrams from Chapter 7 that I'm going to refer to, and then some diagrams from another textbook because I like the way that it's labeled. So let's get started. So aerobic respiration, well, first of all, before I start with that, as a reminder, what is metabolism? And metabolism, that's the word that we use to describe all chemical reactions that happen inside of a living thing. So whether you're one cell big or you are more than one cell big, all of the chemical reactions inside of you are referred to as your metabolism. Remember that metabolism has two parts. There's the part that breaks things down, your catabolic reactions or catabolism, and then there's the reactions that build, or the anabolic reactions. So these, they tear apart and these build. Other names for these types of reactions, you can call them digestive reactions, or you can call these exergonic reactions. Anabolic reactions, you can call them, this is a fancy word, biosynthetic. And the word biosynthetic literally translates into life building. So biosynthetic is kind of the opposite of digestive. The opposite of exergonic would be endergonic. Now, a quick thing about chemical reactions in general. When you have a chemical reaction, it's divided into two halves. You've got some stuff on this side of the arrow pointing away from the arrow, and then you have some stuff that the arrow is pointing to. The stuff that you start with, your starting material, those are called reactants. The stuff that you produce are called your products. So for example, if we talk about the digestion of starch, starch can be broken down into many individual glucose molecules. So this type of reaction, starch is the reactant, the products are many, many glucose molecules. Now, starch itself is made up of all these tiny glucose molecules that are linked together by chemical bonds. So when you digest the starch, you're breaking the chemical bonds. When you break chemical bonds, you release energy. So digestive reactions release energy. The word exergonic literally means energy out. So if energy is a product, then you have an exergonic reaction. This energy then becomes available for the cell to use. Well, we can reverse this reaction like many plants do, and they will take several glucose molecules and link them together in order to create a large starch molecule. So I'm just going to represent starch as all these individual glucose molecules linked together. And these little lines right here represent a chemical bond. So this diagram has, several chemical bonds in it. Here's my point. When you create chemical bonds, what you're doing is you're using energy to create the bonds. Chemical bonds store energy. So if you take these individual glucose molecules and you're going to link them together, you have to have energy to do that. So if energy is on the reactant side, That's what we call an endergonic reaction. Endergonic reactions are literally energy building, whereas exergonic are literally energy releasing. So what we're going to find in metabolism is that we couple a lot of reactions, meaning we put reactions together, some that are going to tear things apart so that we can get the products that we need in order to put other things together. A good example of that is going to be the ATP cycle. So ATP stands for adenosine triphosphate. That's the energy molecule of all cells. It's made out of an adenosine molecule and 3 phosphates linked together. The third chemical bond is usually drawn as a squiggly line because there's a lot of energy stored in that third bond. If I break this bond right here, I end up releasing energy. So the breakdown of ATP releases energy. What you get left over is your adenosine, but this time only 2 ATP or 2 phosphates are linked up, and you have 1 phosphate that's all by itself. We've broken that chemical bond. An individual phosphate is sometimes called an inorganic phosphate. This thing that's left over is no longer adenosine triphosphate. There's only 2 phosphates on there, so we call it ADP, or adenosine diphosphate. If I want to link these back together, I'm going to need to put energy into the system. So energy in will literally recreate the bond to make our ATP again. So metabolism is a constant cycle of breaking down ATP for the cell to obtain energy. And then when the cell needs to store energy, taking these 2 compounds and making ATP again. Now you're probably wondering, what does a cell do with the freed energy? Well, this energy is going to be available to basically make your muscles move, make your nerve cells send signals. It's going to provide the energy that you need to blink your eyes, to take your next breath, or to literally move something from one side of the cell to the other. So breaking this bond releases energy to do all of those things. Whenever you have to store energy for later, like making fat cells or whatnot, you're going to recreate those chemical bonds to link them together. The nice thing about ATP is it's a readily available energy source. If you have something like a fat molecule, sometimes you have to go through more than one step to break down the fat molecule to eventually get some ATP. But I mentioned fat because a lot of people understand that fat is basically stored energy. It's a long-term energy source, not energy for right now, but energy for later. So my point is, a lot of metabolism uses, a lot of metabolic processes are geared to keep this cycle going. So what we're going to talk about now is basically 2 big pathways or 2 big sets of chemical reactions that we undergo, or all living things, not just us, but that living things undergo in order to keep this particular cycle going. And so an overview of metabolism looks like this. There's a process called glycolysis, which is literally the breakdown of glucose. That happens as a first step. What follows glycolysis depends on how much oxygen is readily available. If you have oxygen present, if it's readily available, then after glycolysis, you start a process called the Krebs cycle. And then after the Krebs cycle comes a series of reactions called the electron transport system. The term aerobic respiration really refers to these two guys together. That's really what is aerobic respiration. Some people count glycolysis as part of this. Really, though, it's just a Krebs cycle and the electron transport, which are considered aerobic respiration, because these things only happen if oxygen is present. And that's what aerobic means, is that it's oxygen is required. Oxygen is required. Respiration, I'll spell it out. Respiration means basically you're taking in oxygen, you're breaking down stuff in order to generate energy. Breakdown of nutrients to make energy. Okay, I'm going to pause the video for just a second. Okay, we're back. So again, quick review. Glycolysis is a series of chemical reactions. Those reactions function to break down glucose. If oxygen is present, then after glycolysis, we undergo aerobic respiration. Aerobic respiration itself is two parts. There's what's called the Krebs cycle and the electron transport system. Well, what happens if oxygen is not available? So no oxygen. Then we have an alternate set of chemical reactions. Instead of doing this, we undergo fermentation. So because fermentation happens in the absence of oxygen, sometimes it's referred to as anaerobic respiration. In Latin or in biology, the prefix "a" in the front is a negator. So it literally means without oxygen. Okay, without oxygen. So what we're going to do is we're going to talk about these steps in a little bit more detail. So here we go. I'm on page 3 here. I need to pause the recording just a little bit. My dog is asking to go outside. Hold on. Okay, here we go. I'm back. So let's look at glycolysis in a little bit more detail. Oh, there goes my dog. We are live, folks. So what is glycolysis? Well, as I mentioned before, it's the breakdown of a glucose molecule. So glycolysis is the breakdown of glucose. So if you can imagine a glucose molecule, it's actually made of 6 carbons all linked together. So it's a 6-carbon sugar. It's a 6-carbon sugar. When we split it apart, we actually split it right here in half. And we use water and a little bit of ATP, 2 of them, as a matter of fact, to split. So remember I mentioned that ATP can be used as an energy source to get work done. Well, in this case, we take 2 ATP molecules and they have enough energy to take a water molecule and literally use it like a little tiny water gun to break this chemical bond right here. And of course, when that happens, the ATP become ADP plus a phosphate and then that goes on to its own sort of thing. The end result of glycolysis is you have two smaller molecules that are 3 carbons each. Each of these is called a pyruvate, or some books call them pyruvic acid. So here's another pyruvate. This is called a pyruvic acid. So pyruvate, pyruvic acid, they're interchangeable. So the end product of glycolysis is 2 pyruvate molecules. This breakdown also ends up generating a little bit of energy. That's the whole reason to break chemical bonds, is to release the energy that is stored there. We actually end up making 4 ATP molecules out of it. But if we consider that we started out with 2 as an investment, if we pay ourselves back, then we actually only netted 2 ATP. So the end products of glycolysis are 2 ATP and 2 pyruvates. Glycolysis, it occurs in the cytoplasm of the cell. It occurs in the cytoplasm because that's where the enzymes that are responsible for this is where they live. So the enzymes that make glycolysis happen, they live in the cytoplasm. Now, if you look at your textbook, I'm simplifying this greatly. Glycolysis is about 6 chemical reactions. You don't have to know the chemical reactions. You just need to know what we start with and what we end with. So you start with glucose, you start with water, and you start with ATP. You also need to know what you end with. You end with 2 pyruvates and 2 ATP. You also need to know where it happens in the cell, and it happens in the cytoplasm. So the starting products, the ending products, and where this happens. So far, so good. As I mentioned a while ago, what happens after glycolysis depends on whether oxygen is available or not. Let's go this direction. We'll assume that oxygen is available. If oxygen is available, then each pyruvate will be broken down even further by the Krebs cycle. Oops, pyruvate. So here comes, I'm just going to draw 1 pyruvate. It's a 3-carbon sugar. This pyruvate enters a series of chemical reactions. How many? About, if I remember correctly, I think it's like 9 chemical reactions, or maybe 8 chemical reactions. And what's crazy is pyruvate gets broken down, and then at the end, it's turned back into a starting product. So you kind of get trapped in this circle where pyruvate gets broken down and then returned back to something that's like pyruvate over and over again. That's why we call it a Krebs cycle. Krebs, named after the gentleman who discovered it, cycle, because you end up making what you started with. Again, you don't have to know all of the parts of the Krebs cycle. Here's what I want you to know, is where does it occur? This occurs on the way to the middle of the mitochondrion. And I'm going to show you a diagram of the mitochondrion in just here a second. But as a reminder, the mitochondrion, this is the cell part or the organelle that makes ATP. Or I think we learned it as it makes energy. So we're going to be eventually making energy, and the conversion of pyruvates happens on the way to where we're eventually going to make that happen. This cycle generates some stuff. It generates a little bit of ATP, like 2 more, so not a whole lot of them. It also generates some carbon dioxide, but most importantly, it generates what are called electron carriers. Electron carriers are not energy now, they are energy later. And the 2 electron carriers that we see in animals are a molecule called NADH and one called FADH2. Both of these are related to B vitamins. They're not exactly the same thing as B vitamins, but they're related to B vitamins. So if you've ever heard that vitamin B is important in your metabolism, it's true. Vitamin D does help your metabolism. These 2 guys are members of that family that help your metabolism go along. They are used as part of this Krebs cycle. So the breakdown of the pyruvates in the presence of oxygen generates all of these things. So these are the end products of the Krebs cycle. The starting products is pyruvate and oxygen. Even though we don't use oxygen, oxygen is the signal for this to happen. And where does it occur? On the way to the mitochondria. So let's look at a diagram of the mitochondrion. I'm going to share a picture here. That's not the picture I wanted to share. This was the picture I wanted to share. There we go. So on your screen, here's a diagram. It's not from your book, but this is, I like this diagram because it's nicely labeled. Mitochondrion is singular, mitochondria with an A ending, that's plural. So here is what mitochondria look like. They have an outside bag, and then they have an inside bag. And the inside bag is folded very neatly, and it's surrounded by the outside bag. So that's why sometimes it's called a bag within the bag. So the bag on the inside is called the inner bag, and the bag on the outside is called the outer bag. You might remember from a previous video that mitochondria are thought to be remnants of bacteria that somehow were incorporated inside of eukaryotes. And when they were incorporated inside of eukaryotes, Mother Nature scooped out the guts, so to speak, of bacteria. She kept the bacterial plasma membrane and then wrapped a eukaryotic membrane around it. So that's why it's kind of a bag inside of a bag. Mitochondria, they're really kind of interesting organelles. If I had to pick a favorite, mitochondria would be it. They do have their own DNA. They can replicate on their own. And the way that their inner membrane works is very similar to how the plasma membrane of bacteria work. So that's where we get this theory that perhaps mitochondria at one time used to be bacteria that Mother Nature kept around inside of a eukaryote because it was useful. So I mentioned that the glycolysis happens in the cytoplasm. So if you can see my arrow moving around, glycolysis happens somewhere around here. Krebs cycle happens on the way to this inner membrane. The last step of aerobic respiration is going to literally happen on the surface of this inner membrane. The reason it's going to happen on the surface is because the inner bag contains special molecules and actually special enzymes. And here's a diagram, a close-up of those enzymes, and they occur in groups of 4. They occur in groups of 4, and those enzymes are going to work with the electron carriers to generate lots and lots of energy. So I'll show you how that happens. Maybe I'll show you how that happens. Oh, wrong button. Okay, here we go. So let me show you how this happens. So what's going to be used in the next step is these guys are going to continue to the electron transport system. They're also going to continue, or they're going to take with them, in a sense, the available oxygen, because now this is where it's really going to become important. So the electron transport system. So I'll recreate the PowerPoint picture that I just showed. Here's the mitochondrion, and here is the inner membrane of the mitochondrion. And I'm just going to draw a group of 4 enzymes. They're studded all along here, but I'm just going to draw a group of them. So glycolysis happens out here, and Krebs happens on the way. And then when we get to the inner membrane, that's where electron transport is going to actually happen. So once our products get here to the electron transport system, we have NADH and FADH2. These guys are, again, electron carriers. They're going to donate their electrons to these enzymes that come out in a group of 4, and those are called cytochromes. These are enzymes in groups of 4 that live on that inner membrane. Now, electron carriers, their whole purpose is to catch electrons. So during the Krebs cycle, when we've been breaking things down over and over again, we're literally tearing apart molecules. When you tear apart molecules, sometimes you generate a little bit of chemical waste, in a sense. And one of the forms of chemical waste is you have all these electrons that are kind of flying off of molecules that are being torn apart. Well, electrons that are all free by themselves, they can be bad for cells. They can cause cell damage. If you've ever heard of the term free radicals, those are enzymes, or excuse me, those are electrons that are not, that are free, kind of bouncing around. And so the purpose of the electron carriers is to catch these guys and use them later. Another word for electron carriers, which you probably hear in commercials, is antioxidants. Antioxidants are the molecules which catch free radicals. Free radicals are useful, though, because we can use them to generate energy, which is what we do in the electron transport system. These guys will take those electrons that were floating around, not behaving, if you will. They will take those electrons and donate them right here. The cytochromes, through some elaborate chemistry, but elegant chemistry, they use those electrons, specifically their energy, because moving electrons is electricity. So we use the energy or the electricity of those moving electrons to generate lots and lots of ATP. How many? Anywhere from 28 to around 32 ATP. It varies. It varies on the cell, but we generate quite a bit of ATP. We also generate a little bit of water as a waste product. So the end products of the electron transport system are ATP and water. This process only happens in the presence of oxygen. Oxygen is actually part of the generation of a little bit of water. It's what kind of makes this whole thing happen. So if you're counting ATPs, we've got 2 from glycolysis. From the electron from the Krebs cycle, we've generated another 2 ATP. And then from the electron transport, we have a range of them. So let's just use an average number of about 32 ATP is considered like the average number that you make. It can be higher or it can be lower, but a total breakdown is 36 ATP from the breakdown of 1 glucose molecule. So if you see a test question or a quiz question that asks you, how many ATP do you get from the breakdown of 1 simple sugar? There's your answer, 36 ATP. Still with me?
Okay, so on an average of the ATP breakdown is an average of 36 per 1 glucose molecule, correct?
Right. The reason it's an average is because some cells do this process better than others. So like, for example, muscle cells are really good at generating ATP. So they will generate maybe 38, maybe 40, maybe 42, maybe up to 48 ATP. Other cells that are not as efficient at doing this, or they don't have as many mitochondria, they might only generate like 26 ATP. So the number 36 is considered like the most common average that people report as being how many you get out of 1 glucose molecule. So on the quiz, or I said like on the test, when they ask you how many do you get, it's going to be 36. And that is from glycolysis and aerobic respiration. Remember, this part is the actual aerobic respiration part. One more thing, and that's going to be what happens if no oxygen is present. So what we just talked about was going through this path to make a total of 36 ATP. What happens if oxygen's not present? How does this path go? Well, we still have glycolysis. It's unchanged. So we still break down 1, 2, 3, 4, 5, 6. We still break down a 3-carbon sugar or 6-carbon sugar into 3. Let me say that again. We still break down a 6-carbon sugar into 2 3-carbon pyruvates. So that doesn't change. It still occurs in the cytoplasm. It still requires a water molecule and 2 ATP to get started, and it still generates 4 ATP when we're done, but since we have to pay ourselves back, we call it 2 net ATP. So that doesn't change. What does change is what happens to the pyruvate if there's no oxygen available. If there's no oxygen available, one of 2 things happens. The pyruvates are converted to something called lactic acid. Or the other option is that the pyruvates are converted to ethanol and carbon dioxide. This process occurs in the cytoplasm. So that means that glycolysis happens in the cytoplasm. If there's no oxygen available, then the pyruvates stay in the cytoplasm and are broken down even more. The difference between making lactic acid and making ethanol and carbon dioxide It depends on what kind of cell that you are. So if we're talking about animals, this is very common in muscle cells. In fact, you and I, this is what we do when we undergo fermentation, is we make lactic acid. Lactic acid is the stuff that spoils milk. So are you telling me that you make the stuff that spoils milk inside of your cells? And the answer is yes. As long as your muscles are warm, lactic acid remains a liquid. But if your muscles get cold, lactic acid crystallizes, and that's what causes muscle soreness. So if you are exercising intensely, or if you are exercising for the first time in a long time, you're going to require a lot of energy and a lot of oxygen to keep up the exercise level. Here we go. This is what I'm looking for. So if you're exercising intensely, you're going to need a lot of energy. So you're going to try and undergo aerobic respiration. But if you haven't exercised in a while, you literally do not have enough mitochondria in your cells to make this happen, or your mitochondria are just not very efficient. You literally cannot take enough oxygen to keep this process going. So some of your cells are going to go through fermentation because they just don't have sufficient oxygen to keep up with what you're requiring of them. If your muscle cells undergo fermentation, then you're going to produce lactic acid. And as I mentioned, as long as your muscles are warm, this stays in solution. In other words, it stays like as a liquid. But when you stop to rest, maybe go to bed for the night, and you wake up the next morning, the pain you feel in the muscle cells is to this being a crystal. Now, I'm not talking like a big crystal, but tiny little crystalline parts. And when your muscle cells warm up, this dissolves in water again, and it alleviates some of the soreness. Eventually, your body will flush this out, but it takes some time. Who undergoes ethanol or alcoholic fermentation? Ethanol, that's the type of alcohol that you drink. Well, yeast cells and also certain bacteria. Alcoholic fermentation using yeast is big business. The entire beer, wine, and spirit industry depend on this process in order to generate their product. So this is how ethanol is made, is that we allow yeast to basically break down sugar in the absence of oxygen. Bacteria, certain bacteria can do this process also. So the take-home message about fermentation is, where does it occur? In the cytoplasm. What conditions are required? No available oxygen. What do you use to start fermentation? Well, you need a little pyruvate. What can you make? Either lactic acid if you're an animal cell, or ethanol and carbon dioxide if you're yeast or bacteria. By the way, fermentation doesn't really generate any ATP. The point of it is to generate some other things that we can eventually use to start glycolysis again. But I promised you a simplified version, so there you go. So on your quiz or on your test associated with fermentation and aerobic respiration, you might see some questions like, where does fermentation occur? What conditions are required? What do you need to start it? What are your end products? Who produces those types of end products? And how much ATP is generated by fermentation? None. I'm going to stop my video here. I will post my notes on Canvas. And those of you that are on live with me, I will entertain your questions. So I'm going to stop my video. Thanks for watching.